NRF2, nuclear factor. Erythroid 2-related factor 2 is one of the most important transcription factors in cellular stress defense. It's the master regulator of antioxidant gene expression, and its activation produces a coordinated upregulation of the proteins that protect cells from oxidative damage. The connection between creatine and NRF2 is one of the newer and more mechanistically interesting areas of creatine research. NRF2 sits inactive in the cytoplasm, bound to a regulatory protein called KEAP1. When the cell detects oxidative or electrophilic stress through reactive oxygen species, electrophilic compounds, or physical cell stress, KEAP1 releases NRF2. Free NRF2 migrates to the nucleus, binds to antioxidant response elements in the DNA, and activates a suite of protective genes. These include genes encoding glutathione synthase, hemioxygenase 1, NADP, H-quinone, oxidoreductase, and several other proteins that collectively constitute the cell's primary antioxidant arsenal. NRF2 activation is one of the most powerful endogenous mechanisms for cellular stress protection available. The creatine connection comes through two pathways. The first involves cellular energy status. NRF2 activity is regulated partly by the cell's energy state, with adequate ATP levels supporting normal NRF2 function. When cellular ATP drops sharply, as it does in intense exercise in unsupplemented muscles, the energy stress compounds the oxidative stress and can impair the normal NRF2 response. Creatine's maintenance of higher ATP levels during intense exercise supports NRF2 function by preserving the energy environment in which NRF2 operates. The second pathway is more direct. Some research, primarily in cell culture systems and animal models, suggests that creatine or its metabolites can directly influence KEAP1-N, RF2 interaction, or NRF2 nuclear translocation. The mechanism is not fully characterized, and the in vitro findings don't always translate directly to human physiology. But the direction is consistent. Creatine supplementation in exercising animals and in some human exercise studies is associated with better preservation of NRF2, target gene expression, after intense training. The practical relevance of the NRF2 pathway extends well beyond exercise performance. NRF2 activation is one of the cellular mechanisms associated with longevity, reduced cancer risk, reduced cardiovascular disease risk, and neuroprotection. Compounds that activate NRF2, including sulforaphane from broccoli, resveratrol, and curcumin, are among the most heavily researched in the longevity and preventive medicine space. If creatine supports NRF2 function through energy homeostasis and possibly direct activation, its health implications extend considerably beyond sports nutrition. This is an emerging area where the evidence is preliminary, but the mechanistic case is compelling enough to warrant serious ongoing investigation. For athletes, the NRF2 angle reinforces what the ROS and antioxidant research already suggests. Creatine's benefits to cellular health during intense training go beyond the phosphocreatine buffer and the protein synthesis signals. The cell protection story is a legitimate secondary benefit that adds to the case for long-term consistent supplementation. The NRF2 research on creatine is part of a broader scientific conversation about nutritional compounds that activate endogenous cellular defense pathways rather than providing direct antioxidant activity. The conventional antioxidant supplementation story, take high doses of vitamin C, vitamin E, beta-carotene to neutralize free radicals, has been problematic. Large randomized controlled trials of supplemental antioxidants have failed to produce the health benefits predicted from the free radical theory of disease, and some have shown adverse effects. The emerging understanding is that moderate ROS production during exercise is actually a beneficial signal. It activates NRF2, mitochondrial biogenesis, and numerous other adaptive responses. Taking high-dose exogenous antioxidants blunts these adaptive signals, potentially impairing the training adaptation. This is the mechanism behind findings that high-dose vitamin C and E supplementation impairs strength training adaptations in some studies. The problem isn't that antioxidants are inherently bad. The problem is that high-dose exogenous antioxidants substitute for the cell's own defense systems, rather than supporting them. Creatine takes a different approach. By preserving cellular energy status during intense exercise, it supports the cell's own NRF2-mediated defense rather than replacing it. The cell still produces some ROS, enough to generate the adaptive signal, but the energy environment in which the defense mechanisms respond is better preserved.